We are discussing mitochondrial mapping, AI, and cellular cartography. Welcome to MitoGenesis, where we unlock secrets of life's smallest engines, our mitochondria, and explore how modern science and technology are reshaping how we understand and manipulate them. Today's episode is about maps. No, not the ones you use to find a coffee shop, but cellular maps. More specifically, maps of mitochondrial behavior, movement, and morphology. You see, mitochondria don't just sit quietly inside the cell. They are dynamic, constantly moving, fusing, dividing, and reorienting themselves, depending on cellular energy demands, developmental stage, or stress. And this distribution isn't random. It's tightly regulated, and in many diseases, especially neurodegenerative disorders and cancer, mitochondrial misplacement is a hallmark. But mapping these movements and identifying dysfunctions in real time across thousands of cells, that was once a fantasy. Now, AI and obviously a lot of associated technology, including imaging, is changing all of that. To break this down, I'm joined with Basudha Banerjee. She's a young scientist who's interested in regenerative medicine and stem cell biology. Over to you, Basudha.
Thank you so much for having me, Dr. Khurana. So, as you begin, you're absolutely right that the mitochondrial behavior is one of the most underappreciated forms of cellular intelligence. And with the current advancements in life cell imaging, we can now watch how mitochondria is in action, how they swarm towards areas of high ATP demand, how they cluster at the base of dividing cells, and how they change their shape during stem cell differentiation. But it is not enough to watch. We need to quantify. And this is exactly where AI powered segmentation steps in. Using deep learning models trained on fluorescent imaging datasets, we can now automatically identify, track, and analyze the mitochondrial networks across thousands of life cells. And these models would go beyond what a human eye can perceive. They can identify morphological signatures of dysfunction, like swelling, fragmentation, or even immobility, even before a researcher might notice them.
Well, fascinating. And how is this used in stem cell biology? I'm especially curious about mitochondrial positioning in stem cell niches and how that impacts fate decisions.
Well, that is a great question and something stem cell biology I love to talk about. So, in stem cell populations, particularly in the embryonic or induced pluripotent stem cells, mitochondria are relatively immature and are perinuclear. And as differentiation progresses, these mitochondria would migrate, elongate, and they become more networked towards regions of the cell associated with a slightly higher oxidative metabolism. And now we are using AI enhanced time lapse imaging to predict where mitochondria will redistribute as a stem cell commits to a certain lineage. So this is very interesting. It's like even during mitosis, AI tools can model and forecast the mitochondrial inheritance patterns, which we now know affect the daughter cell viability and function. And most importantly, I would like to mention that the AI guided mitochondrial reprogramming. This is one of the most emerging frontiers where we can track not only the nuclear transcriptional changes, and but also the mitochondrial behavior, the associated oxidation levels, and membrane potential across the entire reprogramming timeline.
We are really talking about AI not just as a tool for observation, but for prediction and manipulation.
Exactly. One very powerful use case is the early disease detection. For instance, in neurodegeneration or diabetes, mitochondrial dysfunction often presents as a subtle morphological shift, fragmentation, like loss of crystal structures, or even a stalled transport. Deep learning models, especially the convolutional neural networks, they're trained on 3D confocal or super resolution microscopy, and these models can flag these patterns before clinical symptoms even emerge. And if we integrate this with multi-omics, say combining single cell RNA sequencing, proteomic data, and live imaging, then we can build a predictive model of cell fate, disease progression, and even therapeutic response. That is the cellular cartography in its truest sense, an integrated AI annotated map of life at the microscopic scale.
Well, you're painting a very powerful vision. The future where AI doesn't just help us see cells, but helps us intervene optimally, early, and precisely. Imagine customizing mitochondrial therapies in stem cell grafts based on AI predicted mitochondrial health, or predicting which neurons will be lost in Parkinson's years in advance based on mitochondrial imaging fingerprints. That's the promise of mitogenesis.
Absolutely. So AI is no longer just a microscopic assistant. It is becoming a cellular GPS, a diagnostician, and even a therapeutic guide. And the better we map the mitochondria, the better we understand not just energy, but the life itself.
Well, and on that note, thank you for joining us. To our listeners, this is only the beginning. In our next episode, we'll dive deeper into how mitochondria remodel during stress and how AI helps us decode their SOS signals in real time. Until then, stay curious, stay mitotropic. This is Mitogenesis signing off. I'm Sukant Khurana, your host, and with me is Basudha Banerjee, our expert. Thank you once again.
Thank you.